welcome to the biomedical faculty candidate seminar in mechanical engineering. Today we have with us Dr. Jacot, if I pronounce it right. He's an associate professor in Rice University. He got his BS in chemical engineering at CU Boulder, then had extensive industry experience, almost 7 years I think. Then went on to get a PhD in biomedical engineering from Boston University, and then a postdoc from UC San Diego. And after his postdoc, he joined Rice University in 2008, and now he's a tenured faculty member there. During his time at Rice, he got many grants and awards, including the prestigious NSF Career Award and more recently NIH R01 Award. Today he's going to be talking to us about biomaterials and how he's going to use those to repair heart defects. With that, Jeff, it's all yours.
Okay, great. Well, thanks to all of you for rearranging your schedule and coming to this.
Thanks for coming twice.
Yeah. So I'll start off with kind of motivation behind the research that I've been working on. And we're looking at how to use novel materials and controlled material substrates and scaffolds, as well as stem cells for the repair of birth defects, focusing primarily on congenital heart defects. Congenital heart defects are the most common birth defect and the most common fatal birth defect. It's the most common non-infectious cause of death in the first year from congenital heart defects. And in addition to heart defects, there's been a big increase in the amount of children going into heart failure. Much of this because of palliative surgeries that really bridge from babies who have initially died very shortly after birth to surviving with some sort of heart defect or extra stress on the heart that ends up going into heart failure. And many types of complex heart failures or complex heart defects require several surgeries over a child's life and don't end up giving full heart function back. So the only complete way to get full heart function is a transplant, and there aren't enough transplanted hearts. So we started to look at how you can use recent advances in regenerative medicine and in biomaterials and tissue-engineered scaffold design to try to make something that can get us from current repairs of heart defects to really a complete repair and complete cure. Right now, the state of the heart for repairing structural heart defects is to restructure the heart with a patch. And this patch can either be on the inside in a septum, repairing holes between the atria or the ventricles, or can open up places on the outside of the heart. And here's an example of one defect I'll be talking about a lot called tetralogy of Fallot, where there's a blockage, a muscular ingrowth that blocks the right ventricular outflow tract, where blood flows from the right ventricle through the lungs. Because of this, there are other defects that develop, muscular right ventricle overriding aorta, a ventricular septal defect. And to correct these, surgeons will cut out that muscular area that's normally contractile conducting heart tissue in the right ventricular outflow tract, and will go in, put a patch in the ventricular septum to stop blood mixing in the ventricles, and then a patch in the right ventricular wall to open up that area so that blood can flow more freely to the pulmonary artery and into the lungs. And the most common materials for this are either Dacron, occasionally Gore-Tex, or fixed pericardium. This is a membrane that surrounds the heart that surgeons will cut through to get to the heart, and can cut a piece out of that, dip it in glutaraldehyde, so it cross-links everything, kills all the cells. So it becomes more like a plastic material, and then washed and put back into the heart. And in all of these cases, what you end up with is scar tissue. So you have this infant who has this defect corrected. And in severe cases, at 2 to 3 months of life, in less severe cases, 9 months to a year old. And starts off with what becomes just scar tissue in an area that in a typical heart is contractile heart tissue. So not surprisingly, this greatly increases the chance of heart failure and the chance of arrhythmias developing. And arrhythmias are very likely to develop because you have a section of the heart that isn't conducting the electrical heart signal. So you have an abnormal flow of the electrical conduction throughout the heart, which can lead to an arrhythmia. So I've looked through how can we, instead of just putting in a piece of plastic, put in something that becomes part of the heart, that either contains cells or contains a material that will just go away and grow with the patient and not just be the scar tissue. I'll start off talking about cell source, and then I'll get into the biomaterials. One of the biggest needs really is cell source. So looked into various sources of cells. I'll talk a little bit about induced pluripotent stem cells, but started to focus on this population of cells in amniotic fluid, and talking to people who deal with heart defects, and very severe heart defects, at Texas Children's Hospital, where my laboratory is located. When you're in a major teaching hospital like that, about 80%, they measure 80% of the defects that either lead to death or require surgery within the first year of life are detected in utero in a routine ultrasound. Usually second trimester ultrasound will detect this so that when the baby's born, surgeons already know they're going to have to do a repair. During this time, looking at if we're going to use cells, what cells might be available, so looking into amniotic fluid stem cells. There's a population of cells in amniotic fluid that is very embryonic stem cell-like, doesn't have all of the pluripotency of embryonic stem cells, but can be made into several different types of cells, and has been cultured and shown to have dural differentiation and vascular differentiation. Dural differentiation, I guess, it was shown to have bone differentiation by other groups. And so we thought if we could get these cells and make some sort of tissue out of these, it would be great because these cells are genetically matched to the developing fetus that's going to need a surgery shortly after birth. So we think that if this is detected sometime in second trimester, we'd come up with some sort of therapy that isolates stem cells at that time, before the baby's born, expands those cells, makes the right shape and patch that's needed of living cells to be used at birth for repair, seeds those cells into that tissue construct, and then when the baby's born, use that construct of cells genetically matched to this newborn infant in the heart repair that that infant needs. And we think that if we do something like that, we can end up with a normal heart. And the goal is to have something that doesn't form a scar, that a few months after the surgery, you can't tell the difference between the implanted tissue and normal tissue. And we think that that not only would increase the, would give better outcomes in current surgeries, but we think that could allow surgeons to start thinking about different surgeries they could do. For example, there are about around 1,000 infants a year in the United States who are born with a single ventricle, usually with a syndrome called hyperplasic left heart syndrome, meaning that the left ventricle doesn't form, and you end up with a single right ventricle. There are some very smaller number of children born with a single left ventricle. Usually it's a single right ventricle, and you could do things like build up an entire ventricle if you had working heart tissue. So with these cells, we've been able to culture human amniotic fluid stem cells. We get amniotic fluid from cases in the fetal intervention department. Most of what, in fact, everything that I'll show in this talk all comes from cases of what's called twin-twin transfusion, with identical twins that share a placenta, where one twin ends up stealing blood from the other twin. And it's one that can, if untreated, lead to loss of both fetuses. And surgeons can go in and laser ablate the blood vessels that are passing between the twins, so that vessels are just coming from the shared placenta to each twin. And to do that laser ablation, they need to take off about 180 milliliters of amniotic fluid, which is a huge amount. And it's really the most amniotic fluid ever taken off in a procedure that they routinely do. And I say routinely, that's done, they do probably about one a week of these at Texas Children's. There are maybe 3 places, 4 places in the nation that really specialize in this. Another place that specializes in this is Children's Hospital, Colorado, just right down east of Denver. So that's an interesting thing with being here. And the good thing about it is these are infants that don't have a congenital defect, whose hearts are typical development. We get all that information, but where we get a lot of cells, so we can do a lot of work on these cells. We've also done work with cells from infants who are diagnosed with a heart defect. In fact, typically, as soon as a fetus is diagnosed with some sort of heart defect, one of the first procedures that will be done is amniocentesis, just with a needle, take off amniotic fluid, so that clinicians can get cells and test them and do a karyotype like this and look for chromosomal abnormalities, because there could be, actually pretty likely that there's some sort of chromosomal abnormality when heart defects are detected. So we do get cells from that, currently just looking at the differences between those. I think that's actually, I'll talk about that in my future projects. I think that's one area we can do big things. But we get these amniotic fluid stem cells. They're karyotypically stable. Here I show over 4 passages. Everything I'll show in this talk today is done at passage 4, so about 20 doublings. Here I show this. I like to show the male cells because this one has X and Y chromosome. So these are the fetus's cells. We've never gotten a cell that we could identify as a maternal cell. It's all fetal cells that we get from amniotic fluid. And we sort these cells for c-KIT positive sorting, a marker of mesenchymal-like and pluripotent stem cells. I'm not sure it's the best marker we could sort for, but most of what I'll show here is sorted for c-KIT positive cells. We're now sorting for some other markers that we think give better cells for cardiovascular differentiation. But if we sort for that, we get about 3% of the total cell population. Most of the cells are epithelial cells because they either come from the amniotic sac or they come, so the amniotic fluid just cycles through the fetus continuously and just sloughs off epithelial cells into it. So if we sort for c-KIT positive cells, grow those up over 4 passages, we find that most of the cells are positive for HLA-1 but not HLA-2. So this suggests that the cells do have to, in therapy, be either genetically matched to a patient or with some sort of immune system repression. So these are best used in a treatment where you're getting them from a fetus, using them in the fetus or the newborn for therapy with their own cells. They're positive for stem cell marker SSEA4. If we passage them 4 passages, we start off with 100% c-KIT positive, end up with 25% c-KIT positive. Positive for all the mesenchymal stem cell markers, and negative for all hematopoietic and endothelial epithelial cell markers. So here I show 2 CD41, 31, CD45. So we're getting that epithelial cell population sorted out. We also do not see any sort of hematopoietic cell population in this. So we've taken these cells, and just in interest of time, I'm not going to go through all the studies, although I can answer any questions on studies we've done to characterize these. We want to look at, first, can we make these into cardiomyocytes? They can express cardiac proteins, but without any sort of genetic manipulation. They have already gone past the point where they can be made into fully mature contractile cardiac muscle cells. However, we have been able to genetically reprogram them into induced pluripotent stem cells. So using a non-viral method of making iPS cells, we put in this mRNA 6F modified mRNA transcription factor cocktail, the four Yamanaka factors, transcription factors, one chromatin remodeling factor that increases the efficiency of iPS reprogramming, and then green fluorescent protein just so we can see which cells got transfected. And we figured out we managed to do this in a non-viral method that works for some progenitor cell types, and it seems to work for cells from amniotic fluid. We get these great embroid bodies. And I don't know if many of you have experience with embryonic stem cells or induced pluripotent stem cells, but one of the gold standards is if we take these cells and we inject them into a nude mouse, they form a teratoma. And if you've looked at histology, this is a really cool picture because we have all three germ layers. So we have mesoderm and ectoderm and endoderm all in one picture, one field of view under a microscope. So we can show that these can become any part of a human, apparently, when in the right environment, and this nude mouse that won't reject them. And so their question, I know much of this is a mechanical engineering audience. I don't know if there are questions on induced pluripotent stem cells at this point. Yes?
I do. Just when the baby's still in fetus and you're trying to culture these cells, like say they know they have a heart. I had a child who had a heart defect. Okay. So how much amniotic fluid do you need to take out? I know for me personally, I was in a bad state, so I wouldn't have said, oh yeah, sure, why don't you make my life worse by taking these. So I mean, how much would you need in order to get enough stem cells to culture onto your scaffold that you're doing?
Yeah, so the calculations we've done, if you take out about 10 milliliters of amniotic fluid, which, unless there's some unusual case with very low amounts of amniotic fluid, shouldn't produce extra risk versus the risk of amniocentesis anyways. You can, by the time the child's born, have enough cells to repopulate an entire newborn heart. Now, you lose some of the efficiency at each step that you're doing that with. So that, it's a little tough to say that that's the exact equation that you need. But I think you could do it with a 10 milliliter.
Yeah, that's not very much.
So we can take these cells and we think they can become anything, and we can show that we can differentiate them, get cardiac differentiation at a very high, relatively high efficiency. We do this, we get about 65% efficiency with a method that does not use growth factors, so we use small molecule inhibitors. All of this, we focus on, can we do this without viruses, without growth factors, without animal products, so we get something that we think if we do get valid therapy, that this is something that is feasible to use in the clinic. So we get that, we get good sarcomere formation, calcium transients, and here's just a movie of a dish of what were amniotic fluid stem cells that have been transformed to an iPS state and then pushed towards a cardiac muscle state. And these are just sitting, beating in a dish without any stimulation at all, just spontaneously beating in a dish. So we've looked at, if you're going to put cells into the body, how do you keep them alive? You need some sort of vasculature. And we focused on how do you get a good vasculature into a patch. In fact, I think, and one of the grants that I have is specific on the hypothesis that in developing pediatrics, especially newborns, you likely only need to make a really good vascularized area, and muscle cells will grow into it. And so the vasculature is, I think, more important than the muscle cells. And the one thing you learn about, now hundreds of studies injecting cells into the heart, in failing hearts or after an infarct. And one thing you learn is almost all of them die. And especially if you try to inject muscle cells into the heart, they tend to all die. People inject mesenchymal stem cells or other progenitor cells. A very small portion of them live. I don't feel that there's any thorough study that's proven that anything becomes muscle cells, but it can stabilize the vasculature. And so the important part, I think, is really getting a vasculature. For that, you need endothelial cells. So we want to know, can we make amniotic fluid stem cells into endothelial cells? And by adding very high levels of VEGF, vascular endothelial growth factor, we can. This is flow cytometry. We're looking for CD31, which is a marker of endothelial cells. And you get into 50 nanograms per milliliter, so kind of super physiologic levels of vascular endothelial growth factor. And you get about 30% of the cells expressing CD31. You see we have kind of this dose response curve up to 50 nanograms per milliliter. It doesn't seem to get better or higher than that. And everything I'll show here is compared to positive control of human umbilical vein endothelial cells. So we've looked at these cells. One standard about, let's see what it says, like in this paper, 9%, we've now gotten that higher to about 20% of the cells that we could sort out a triple positive for VE coherent, VEGF receptor 2, and CD31, which really categorizes them as vascular endothelial cells. So the type you would find lining blood vessels. And I want to put this into something that's angiogenic. One issue is that you can throw these cells into matrigel. They form these great little networks that look like capillaries, but if you cut through them, they don't have open lumen. And I don't know if any of you have worked with matrigel. This is a product you can buy from BD Biosciences that's made by this mouse, I think it's a liver cancer line that just spits out tons of extracellular matrix, which cells love to grow on, but it's not anything you could ever use clinically. So we wanted to find something that we can use clinically that induces vascularization, and that induces vascularization in the sense that you do get open lumen, that maybe we could make a pre-vascularized patch. And we found that fibrin, so fibrin is a component that makes blood clots in the body. Starts off as inactive fibrinogen, and when that fibrinogen encounters thrombin in the presence of calcium, it cleaves the fibrin, fibrin self-organizes, it can form a blood clot, it can also just form a gel that encourages blood vessel growth into it. You take these fibrin gels, you can get open lumen through them with endothelial cells. However, the endothelial cells, if you put them at a high enough concentration that it would really be physiologically feasible to use it, they also secrete proteins that just chew up an entire fibrin gel. And so you don't end up with a tissue, you just, if you're only using vascular cells, you just kind of end up with mush. And we wanted some way to stabilize this, so started looking through it, how we could do that, and ended up stabilizing it by reacting polyethylene glycol that's kind of an inert biomaterial along fibrinogen. We can then react it. We make these gels. You can see here a fibrin gel is cloudy, a fibrin peg gel is clear, which makes the microscopy really easy. With very fast gelation times on the order of seconds, and weight retention if you put in enough vascular cells to really provide oxygen flow through one of these, blood flow through one of these, you get weight retention where you actually still have a physical material. What's going on here?
Apparently we're not in the room.
Yeah.
This is good. Yeah, actually.
Yeah, this is good for this slide. Perfect.
So true.
So we can put that in, and I can show you these top slides are in vitro, and you can, all of this is published, but I can answer your questions about it too. You put it in, you form these capillary-like networks. If it's just endothelial cells, you form capillary-like networks in a week, and then they fall apart by 2 weeks. Found out if you add in a 4-to-1 ratio, just unfractionated amniotic fluid stem cells. So these are stem cells, they act like mesenchymal stem cells, and they can turn into these smooth muscle-like or parasite-like cells that can surround blood vessels, and it seems to stabilize the blood vessels. So if you add those, then you get blood vessels that after 2 weeks are stable. You can cut through, this is just a stain, you cut through the gel and you can show that you have fibrin on the outside and these hollow lumen that form with just liquid on the inside. We stain red for CD31 and green is alpha smooth muscle actin. And you get what looks like a native capillary blood vessel with endothelial cells that are stained with CD31 and smooth muscle-like cells that are stained with alpha smooth muscle actin supporting on the outside. We could take these, put these in a nude mouse model. This is just a subcutaneous implant on the back of an athymic mouse. And if we do that, within a week, we get the cells inside survive and the vessels anastomose to the mouse vessels. We can image them. We get, this guy came out a little bit poor quality, but we get red blood cells inside of the gel. And we get the same staining endothelial cells lining an open lumen with support from smooth muscle-like cells. And I call them smooth muscle-like cells because we can also show that these really aren't vascular smooth muscle cells, but they're more kind of pericyte cells supporting and stabilizing the vessels. So this is where I think we have a cell population that can be used to pre-vascularize something, that can quickly hook up to the host vasculature. And if we have this in the right environment, maybe have a patch that we can implant in the heart that's at least in an infant with extra regenerative capacity can be taken over by muscle cells and blend into the rest of heart tissue. Now, one thing that as mechanical engineers, I think this group would find really interesting, we've looked at what mechanical environments. So we want to put in cardiomyocytes, we want to recruit immature cardiomyocytes. What makes cardiomyocytes mature? There are many studies on different growth factors that in other environments that make cardiomyocytes mature, laminin and other extracellular matrices, but you still don't get, in fact, you don't get cardiomyocytes that look like they look in the body. And started to look at how does force and how does the mechanical characterization of the extracellular environment really change these going from an immature cardiomyocyte to something that's mature. Also kind of motivated by some studies that I've done, looking here in mouse development, this big change in elastic modulus from an immediate synthesis of collagen that occurs right after birth in a mouse model. You can also look doing an infarct model tying off one of the coronary arteries in a mouse, let a scar develop, and you get a much bigger elastic modulus change in that scar area. So the mechanical properties are both different as cells are maturing in a mouse model at least, and different in areas after a heart attack or an infarct. Our model system is neonatal rat cardiomyocytes. So you take these cells from neonatal rats, from one-day-old rats, that are immature cardiomyocytes. So they can still attach and spread. You could show by gene profile that they aren't mature yet. And we thought in the right environment could mature over the course of the next week like they do in vivo. So we take these cells and plate them for a week on polyacrylamide hydrogel and can control the elastic modulus. This is shown as a compressive modulus of the hydrogel. If you look at compressive modulus of a native heart muscle tissue, you're in this range of about 10 kilopascals. 1 kilopascal is close to unloaded, the cell's really contracting against its internal cytoskeleton. 50 kilopascal, the cell really can't generate enough force to deform its environment. And the interesting thing is, these cells look a lot like they do in the body. If you just take a cardiomyocyte, image it in a section of the heart. These cells look a lot like cells that you see in papers studying cardiomyocytes that put them in a petri dish. And they don't look anything like cells look like in a body. And they have these stress fibers, which is really interesting. It's actin stress fibers, so big bundles of actin that aren't, that you don't see physiologically in cardiomyocytes. So we know that there's a difference when you put them in different environments. Went into this, started looking at, this is a movie. That's fine. Get these cells that are sitting on top of these substrates and beating in a petri dish. We're stimulating them so we can get an exact beating frequency. And you can get cardiomyocytes that beat. They've been growing on these cultures for a week. And when this beats, it deforms the environment around it. I can measure, so this really comes from measuring the exact shear modulus of the gel. So you know how much shear is required to move these hydrogels. And the mathematics aren't that complex. I actually managed to get from a library this book published in 1880 or something, which had calculated all of the mathematics. Boussinesque is the author. It calculated all of the mathematics for point functions moving on an infinite elastic half space. You know, the shear modulus of the infinite elastic half space. How much shear